Okay, hello. How is everyone? We'll be starting in just a moment. It takes about 30 seconds or so for participants to move from the waiting room into the live stream. Okay, great. Let's get started. So welcome to the HNRCA Monday seminar. Today is our last seminar of the semester. And as I can be seen as the face of the seminar, I'd like to acknowledge the team behind the scenes who make this seminar successful. Eleanor Davison sets up the webinar and provides event support. Mike, Greg, and Andy from TTS are on the AV team. Jose Ordovas and Andres Artisan Korat help in selecting speakers, and Peggy Connolly acts as a liaison with the speakers and coordinates all the seminar activities. So thank you to the committee for all your work, and a personal thank you. I enjoy working with you. We aim to vary the topics over the series, and today our speaker is a leader in the field of aging. Dr. Pankaj Kapahi is professor at the Buck Institute for Research on Aging, and he studies the role of nutrition and energy metabolism in lifespan and age-related disease. The overall goal of the Kapahi lab uses model organisms to study various physiological and molecular processes and their influence by nutrients to impact health and survival. Dr. Kapahi's company, GLYcation, produces a supplement for detoxifying advanced glycation end products. He has been recognized for his scientific excellence with numerous awards in the field of aging. Thank you so much for being with us today. I'd like to now welcome our speaker, Dr. Pankaj Kapahi.
Thank you very much for that kind introduction. I wanted to just say that I got excited about dietary restriction when I was a teenager, and when I first learned about it, I was like, wow, it's amazing that there's an intervention you can sort of participate in, and then if you do this right, you could get an extra decade of life and productive life. So it started my journey in the field of aging, and I was like, this is worth studying. And surprisingly, what I find is that it's much easier said than done. And despite our increasing knowledge about nutrition and dietary restriction, if you look at the public health records, like there are more and more people becoming overweight and obese. We have almost 2/3 of the people overweight. So I feel that there's a lot of potential in sort of understanding and utilizing this knowledge for public good. And I'll sort of take you through my journey since I started and where we are going in this area. So I just want to declare my conflict of interest. As Catherine mentioned, we started a company based on some of the work I'm going to show you as well. So why study dietary restriction? Initially, I have to say it looked very promising in that one idea was that it's very robust because multiple species show a similar phenotype. And one thing I want to point out, though, is though a phenomenon like aging or an intervention can show extension, let's say, of lifespan across species, often people have said it means it's working through conserved mechanisms. I don't quite agree that it necessarily means that it's conserved mechanism as we are seeing, but nevertheless, I think different species, when use some form of adaptation, when nutrients are limiting, to sort of give it this extended time to survive, so that when nutrients are plentiful, it can then revert back to this sort of normal reproductive phase. So I think though we may find different, bacteria, yeast, worms might use in some cases different mechanisms, but I think overall there's also been a lot of success in showing that many mechanisms might indeed be common, and one of them being TOR that we discovered when I was doing my postdoc. And I want to say generally, just if you look at the different genes that have been identified that are important for dietary restriction, they fall in these categories, 3 categories. One is they are sensors, so anything that influences response to nutrients. that can be as a sort of a sensor. Those genes are relevant for sort of influencing lifespan and dietary restriction. So, for example, in this category are sensors for insulin, like IGF, and also TOR, and also potentially sometimes amino acid sensors. And then you've got these regulators downstream, which include transcription factors like FOXO, which is several studies have shown that it's also polymorphisms in FOXO are tied with longevity, and also some of the one I'm going to be talking about are like transcription factors that influence circadian clocks. And then I think this is the part of the puzzle that remains most poorly understood. I would say we're still trying to understand what are the most sort of proximal damage, macromolecular damages that take place that influence aging. And I'll talk a little bit about advanced glycation end products, which is one of them that we think is at least responsible in part for influencing aging. And generally the idea is that you switch from, this is all very well sort of explained also by what's called the antagonistic pleiotropy theory, which is when you move towards a more from an anabolic to a catabolic state, you actually promote you extend lifespan. Whereas if you're in more catabolic state, you can be, as this shows, there's more breakdown and there's more repair, and the animal tends to be in a more youthful state. Whereas in this state, an anabolic state, you're actually more reproductive. But you're also aging faster, but you're more reproductive. So this has been summed in more detail in this recent review we wrote, which I highly recommend to read if anyone's interested in this field, on evaluating the beneficial effects of dietary restriction. And one of the things we did in this review is we started with some of the normal pathways that have been identified, like I mentioned, as sensors, regulators, effectors. But we try to pay more attention to some of the exceptions in the field where this may not. Some of the pathways may be more species specific, or also explaining which ones are conserved. Also, another interesting part of all this puzzle is, like, does dietary restriction benefit all sort of health span measures equally? Does it extend lifespan or elsewhere? And we discussed that quite in depth in this review, and then which sort of gets to this point of that we need a sort of precision medicine approach to sort of understanding this, which I'll get into. So what we initially showed when I did my work as a postdoc in Seymour Benzer's lab is that you inhibit TOR, you can you extend lifespan, and dietary restriction. I mean, the idea would be when the nutrients are limiting, TOR being a key nutrient sensor plays an important role in mediating this effect, and at least this was true in flies. And we also showed that this was in part due to reduction of overall mRNA translation, but to our surprise, some mRNAs are actually getting translated at a higher rate. So we think we found that they will actually, like those important for nuclear encoded mitochondrial genes, actually got translated at a higher rate, and we try to understand why that was, and they were critical for the lifespan extension. Then we later sort of reasoned that this is maybe part of this metabolic adaptation that takes place on dietary restriction. So the animal shifts to a more from using glucose to actually using triglycerides, and for this metabolic adaptation you need improved mitochondrial function. So this we showed in a couple of papers after that. One of them, by Subhash Kateva, who actually showed that the fat turnover, interestingly, on dietary restriction, both in mice and flies, fat turnover increased. Which means, so instead of the animal using glycolysis now, they're actually storing the food as fat, and then they are there's increased turnover of fat, which is utilized by the muscle to keep the animal more active, and that this turnover actually becomes critical for the lifespan extension. So if you block fatty acid oxidation, for example, you don't live long upon dietary restriction. And we also showed that in addition to this metabolic shift, you also need a behavioral shift. And we asked whether if the animal was made immobile, would it still live along the same way? And we found that at least part of the lifespan extension is dependent on the animal also being active. Something we think might be relevant also in humans, like if you're doing dietary restriction, it's one of the, I think, an interesting biomarker to kind of think of. Is it making you more active? If you're more active, then I think that's generally going to be beneficial. But if you push your dietary restriction so much that you become inactive, I'm not sure how well that will translate. So the next question we asked was, how is it that there's both an increase in fat breakdown and fat synthesis? And we reasoned that this could, in part, this could be due to the role of circadian clocks. So as you may be aware, that when you're during the waking period, when we're eating, the circadian clocks evolved so that we could feed during the day when light was present, and then at night we would sleep. And it's different for some animals that are nocturnal. But what's important to note is how metabolism changes during these different periods. So during the day, you're actually synthesizing the lipids and the fat, and these tissues, different tissues like muscle, fat, and liver, they're taking up the nutrients. Whereas at nighttime, when you're not eating, the animal's more in a catabolic state. And we think that the cycling is very critical for also reasons I just mentioned, but it also becomes very relevant at the level of the whole circadian 24-hour cycle. So to investigate this further, we use the fly, and as you know, recently, a few years ago, the fly, the Nobel Prize for work on circadian clocks was given to work that was initiated in fly by actually my mentor Seymour Benzer. They discovered the first gene called clock, which influenced the circadian cycles in flies. And what we asked here was whether when you put the animals under limiting nutrients, does that influence the clocks? And the answer is yes. That under conditions where the animal has low nutrients, a lot of the number of circadian what I call the core clock factors, timeless period, clock, and cycle, you see them actually, you see increased expression, as you can see here. And what's important to appreciate is that there's actually increase in the amplitude of the expression of these clock genes. And in dotted lines are the genes on a rich nutrient diet, whereas in solid lines are the dietary restricted animals. And next, we showed that this increase in amplitude was not just a biomarker, but that this has functional consequences because it's critical for increasing the fat metabolism of the animal, and that this increase in fat metabolism and this increase in gene expression was necessary for the lifespan extension you get under dietary restriction. And just to show that this is quite relevant across species, this is a really nice graph showing respiratory exchange ratios in mice. So now mice, the caloric restriction is done using a different method, but what is common between the flies and mice is what I was just describing you, how the animals shift metabolism. So respiratory exchange ratio of over 1, or 1 means that you're using glycolysis, and over 1 means you're actually synthesizing lipids. So if you see here, you'll appreciate that the animals on caloric restriction, you can see here, they synthesize lipids, and then they're breaking down lipids, and their RQ goes 0.7, means you're actually using fat as an energy source. But look at what's happening in these animals on the ad lib diet. They maintain their RQ around 1, which means they're just using glycolysis primarily. And this is, we think, an important shift that takes place, which is important for explaining the sort of changes in metabolism, the sort of reprogramming of metabolism that takes place. That's important for dietary restriction. And I want to say, recently, some really nice papers, one from the Takahashi lab and one from another lab has come, where they show that in normal restriction of calories, what's also important is this aspect of time-restricted feeding. There's most of the times when people do caloric restriction in mice, Dudley Laming's lab, this is a paper from his lab showing that when the animals are in caloric restriction, they actually eat their food pretty quickly, like within 1 meal. So a lot of what people were studying before as caloric restriction is also a combination of restricting calories, but also eating sort of a time restricted feeding. That means the timing is also important, which means they're giving themselves almost a 20-hour break of not eating. And they did this really elegant experiment to ask, well, what if you ate the same amount of calories, but you ate it multiple times in the day? And what they found was that they didn't benefit the lifespan extension the same way as eating once a day, like normally a mouse does on caloric restriction, and then takes a 20-hour break. So this is further reason to think that, sort of this is an important advance in the field of caloric restriction, because it argues that it's, in addition to restricting calories, it's also important to think about the timing of when you're eating, and maybe eating fewer meals in a day might actually be beneficial. And I have to say, this is kind of surprising if you think about it, that we've been on this planet so long, and we still don't know how many times a day we should be eating for optimal health. So we studied this further in flies, and what we found was that we compared the circadian transcriptome. So now, instead of looking at genes just at one time point, we looked at genes at 6 different time points, and we found that there are more genes that are rhythmic under dietary restriction condition compared to ad lib conditions. And we found that a lot of these genes are enriched for light response. So I'll just break this down. So we looked at gene expression at different times of the day, which is shown here. What I want you to pay attention is that, like, what's happening at different times. So this is early in the morning. We found that a lot of genes important for perception of light would go up under dietary restriction, which is surprising. I never thought that when you eat less, that it would influence your vision, actually. Then middle of the day, genes important for synaptic plasticity and circadian responses go up. And it's interesting, at nighttime, a lot of genes important for inflammation go up. And this is sort of also well known even in humans, that inflammation response actually goes up after the evening. And if you have a fever, you'll see it actually peaks in the evening. And then interestingly, the genes people are most interested in the aging field related to sort of proteostasis or oxidation, antioxidants and oxidation reduction processes, they actually come in the middle of the night. And this is very important to sort of appreciate, because often people will do their experiments during the middle of the day, and you're actually missing out on a lot of changes that may be happening. So I think if anyone is looking at gene expression, do think about when you're measuring the changes in gene expression, because different genes come up at different times of the day, and it's important to consider the timing of the day when you're looking at gene expression. And almost 30% of the genes are believed to be rhythmic. So your gene could very well be rhythmic, and you might be studying it at the wrong time. So we took this study further, and Brian Hodge, who was the driver of this project, showed that many genes that are important in the phototransduction cascade in the fly are actually increased. So this, I won't get into the details of this, but as a photon is sensed by the fly eye, this is very well studied in the fly. A rhodopsin turns into meta-rhodopsin, and then through a kinase, a cascade G protein coupled receptor cascade, it lets in calcium to activate the cell and to sort of transduce the visual signal to a light signal to an electrical signal. And this is then sensed by other cells and transferred. And this, what's interesting is, under dietary restriction, that these animals show an increase in a number of genes in this response. And what we think is happening is that this is actually a very fast happening sort of signal transduction cascade. All these proteins have to get back into their position after like 10-15 minutes. And we think that dietary restriction sort of improves the homeostasis of the whole response, so that light is sensed in a better manner. So what we found, what Brian found, was that if you inhibit genes that are important for this sort of recycling in the eye, that not only does it cause photoreceptor degeneration, but it actually also shortens lifespan. And he showed that this happened because the animals show more inflammation. And then furthermore, he showed that this whole response in the eye, the visual response, is controlled by the transcription factor clock. It seems to be the primary driver. Furthermore, something interesting I want to add is that when we remove genes, inhibited genes that are the rhodopsins, that primary sensors of light, you can actually extend lifespan. Also, keeping the flies in the dark extended lifespan, which sort of fits with this idea that we have, that's we put together here, that under dietary restriction conditions and under dark and these conditions, that the animals have improved sort of reduced phototoxicity and improved visual health of the visual function, which tends to extend lifespan. And this is primarily happening due to reduction in inflammation and breakdown of this eye-brain barrier, we think. Okay, so the next part of the talk is on the role of sort of natural genetic variants. So in the fly, we have used a sort of a very interesting collection of strains, which is described by McKay, where there are 160 odd different strains. And instead of studying flies in sort of one dimension in one strain, we asked the question, okay, what happens when you look at dietary restriction in 160 different strains? All these strains are fully sequenced, and many phenotypes have been studied using this. So we use this to study many phenotypes like body mass, starvation resistance, triglycerides. And what's very interesting is all that I showed you before, things are not universally true. That means in some fly strains, you will find that they can extend lifespan, but this whole response to improving sort of fat metabolism is not necessarily required, but it can still extend lifespan. Or in some cases, you don't see a lifespan extension. So not all strains benefit equally. So we've done several studies on this, and what I want to show you is sort of a snapshot of some of the work. So in addition to lifespan changes, which like this, which is on, so if you look in gray bars, you'll see these 160 lines. Some of them actually shorten lifespan, but a number of flies, we found that about 70–80% of the flies extended lifespan, but to varying degrees. But that's sort of very useful for us because, as I said, they're fully sequenced. So we can now associate what genes are, what polymorphisms are associated with these long lifespan extensions and which ones are sort of driving the lifespan shorter. But the question we asked was, well, is health span equally improved upon dietary restriction? And to our surprise, we used a measure of sort of climbing ability, physical activity in the flies, and we asked, is climbing ability, does it correlate similarly to lifespan? And the answer is no. So a lot of these flies that are living long upon dietary restriction, you can see some are doing well, both in terms of their physical activity and also their lifespan. But many flies, you can see here, they'll actually be physically active, but they'll die shorter, or vice versa. So we think there isn't a one-to-one correlation in this sort of improvement in health span and lifespan. And this is important to appreciate when you think about it at the level of the individual. You all may have noticed that there are a lot of people around you, by the time they're in their 90s, some fall apart more physically and while they're mentally very sharp, and vice versa. So I think I feel going forward, I think a lot of studies in aging have to sort of take into account more the individual variation, because that's a very important factor in what is going to benefit one person over the other. Similarly, drugs like rapamycin that have been shown to extend lifespan also show huge individual variation. So I think we need to think more about this as we go forward. And like I said, this review, I suggest to look at this review. This is also true about dietary restriction. If you look across mouse strains, not all functions in different tissues benefit equally. There's actually a reduction in, for example, wound healing and exercise capacity in some studies, and even weakening of the bone, whereas overall a lot of functions are improved by dietary restriction, like memory and gut barrier function, muscle, but and also sort of liver functions are improved, but there are some functions that don't benefit equally. So we've been paying attention a little more to now what's happening to brain aging under dietary restriction, and like I said, there's widespread improvement, but we don't know the mechanisms, and we think the mechanisms in different tissues are going to be going to end up differently. Yeah, so there's a question, what would the effect of vitamin A restrictions on rhodopsin content? I think, to my knowledge, someone has done this experiment in the fly, and I think it definitely influenced vision, but it's a good question how that would influence the eye function. Because of perhaps increased photoreceptor degeneration, there could be a decline. So on one hand, it's a very tricky thing. You want to, for example, think about this, that a number of long-lived animals, including bats, naked mole rats, actually have reduced visual function and they live longer. But I think the key thing is their eyes are not degenerated. If you do make manipulations that lead to eye degeneration, potentially like vitamin A inhibition, then you could see a detriment to the overall system. And there's also, I want to add to my last part of the talk, that there's also a large study done in like over 5,000 people, I think it's been done 2 times, where they found that people who have increased risk of sort of eye diseases, in a subset of those people, they have much higher risk of multimorbidity. So overall, the idea is that eye disease may not just be a segregated problem, but that you could influence multiple diseases together with it. So like I said, we were looking at brain function. We looked at these different DGRP lines, and we found one gene in particular that caught our eye. It's called mustard. I'll get into it in a minute. You can see that if you have the G allele present, the flies have a higher median lifespan compared to the C allele. So what is OXR1 or mustard? So it was discovered in bacteria to protect against oxidative stress, and that's why it's called OXR1. Overexpression of this in mice seems to be protective in sort of models of ALS. And interestingly, genetic variants in humans are associated with a rare disease where these kids go through cerebral atrophy and they die by almost in their teens. And reduced OXR activity was shown by our collaborator, Hugo Bellin, to cause lysosomal overactivity and brain degeneration. And if you look at the cells, they actually look like this. These dark sort of lysosomes accumulate in these cells. So we were like, okay, what is this got to do with aging? So when we knocked down this gene, OXR, we validated it, and you can see in the dotted line. So in red is the control on rich diet, in blue is the diet restricted animals, and you can see when you knock down this gene, you can actually see a shortening of lifespan, but more specific to dietary restriction conditions. We also got a null mutant of this fly, and you can see normally the flies will again live. Normal flies show large extension of lifespan on diet restriction, but these short-lived flies show almost no extension in lifespan upon diet restriction. So we try to understand what this gene does, and we ask some questions bioinformatically, and it seems like it's important for endocytosis and protein trafficking. A number of genes that are altered seem to be co-regulated with that. And just to summarize this in this slide, what we think is happening is, and I'll get into sort of defining this molecular function. We think OXR1, generally the endosome, what is involved is in sort of receptor recycling of certain genes that are maybe present on the membrane. So not always do you want to break down the gene in the lysosome. You may want to take through the early endosome, you have 2 possible fates. Some, if they're damaged, they go to the lysosome, that would be preferred, but sometimes it can go into the late endosome and actually get recycled. So many proteins get recycled like that, and one can think this recycling could be beneficial, especially when nutrients are limited. But what's very interesting is like what determines what cargo is going one way or the other, and how do different cargoes have different fates. And for that, what's being discovered is this retromer complex, which has multiple components. It's a very dynamic complex, which seems to decide the fate of these proteins that are getting recycled. So also, what's interesting is these genes, VPS 26, 35, 29, have been linked with neurodegenerative diseases in humans. So what we found was, there was also a drug that could stabilize this retromer function. So we said, okay, let's see what happens to our flies that are living short, like shown here. What happens if we give it this retromer stabilizing drug? And you can see here that when we give it this drug, we can rescue the effects almost completely, and even maybe extend lifespan a little bit on the normal conditions. So stabilizing the retromer can totally rescue the OXR1 knockdown function. So we think this OXR may be in part due to its inhibition of retromer function. We also got fibroblasts from these humans with this rare disease OXR1 deletions. And you can see here that, like was shown before, there's an increase in this lysosomal expression in these patients which had the OXR1 deletion. But when we added the retromer, the R55 drug, you can see that overexpression of that lysosomal phenotype disappears. So we think that in part this is mediated, that both in humans and flies, there's a deficit in the sort of retromer function, which leads to the increase in lysosomal numbers. And we also showed that these OXR1 co-localizes to these retromer proteins, VPS35, for example, you can see here. And we've now shown this using IP as well, that the OXR1, when you pull down OXR1, you get other retromer components with it. So the next thing we asked regarding this protein was, does it play a role in sort of neurodegenerative diseases like Alzheimer's? So we collaborated with Nicholas Seyfried, who's got proteomics, really interesting data set, proteomics data set from over 5,000 patients. I'm just checking, I'm still on, I think and they looked at what's happening to OXR1 gene and related genes. So we created what's called a sort of OXR1 network gene network, which means genes that are co-expressed with OXR1. And when we looked at these genes closely and compared them with other data sets in humans, so we find that a number of genes that are in this OXR network actually going down with age. Furthermore, they're actually going, they're also altered in sort of Alzheimer's disease. And interestingly, a subset of genes seem to be driving both AD and sort of being influenced with age. And this subset of genes, as you can see here, linked are important in Alzheimer's disease, Parkinson's, Huntington's, and even a couple of other diseases. So now I'm just showing you some data from the proteomic data set that from Nicholas Seyfried, where we look at the expression of the OXR1, and you can see here they compared control patients with AD, asymptomatic AD, and patients with symptomatic sort of memory loss and in AD. And you can see OXR levels are generally found to be significant, seem to be lower in these patients. And then we so we said, okay, let's see if this would be relevant in a model of tauopathy in flies. So in flies, you can this is the eye of a control fly. And if you overexpress a human tau variant that is pathogenic, you can see degeneration. You see what's called a rough eye phenotype, which essentially means all these cells, some of them are degenerating. So you see this sort of rough eye. But when we overexpress OXR1, you can see that we can largely rescue this phenotype. So it gave us hope that what we're finding in terms of the role of OXR1 may be conserved across in flies and humans. So just to sort of end this part of the talk, wanted to show, say that the summary of this talk is that under diet restriction, we find that there's a transcription factor I didn't get into called TJ traffic jam, which seems to be upregulated, which drives the increased expression of OXR1. And I want to say this is really interesting because some variants of OXR1 go up and some variants don't in the fly. So we found that traffic jam regulates this increase in expression of certain variants, which seem to be improving retromer function. And this retromer function is important in a dietary restriction because it allows recycling of the endosomes. And we think this might improve homeostasis under nutrient limiting conditions. Because if we inhibit OXR1, you see this increase in lysosomal number because the burden of, there's too much burden on the lysosomes now. And what I also showed was that if we use a drug that stabilizes the retromer, you can protect flies from loss of OXR1. And we think maybe OXR stabilizing the retromer will extend lifespan. I didn't show data. Overexpression of OXR1 leads to robust extension in lifespan, and also the R55 drug in certain conditions seems to extend lifespan and also protect against neurodegeneration. So just to sum up this part of the talk, what I said is that we think we need to be sort of thinking more in terms of a precision medicine approach when we think about how diet-based interventions and pharmacological interventions are going to sort of improve health. And I think there are a number of factors that drive this variation, including sex, genetics, epigenetics, and age, which is all covered in this sort of review that I mentioned. And I think we need to be thinking of what biomarkers we need. And it's going to differ on what output you want. You know, some people, they're also noticed, you know, there are very big differences in what people want to achieve. Some people want a better physical ability. Some people want to be, you know, cognitively improved. So I think different biomarkers, different endpoints are going to require different sort of thoughts into what might optimize that sort of health span function. Okay, but so in the last part of the talk, I'm actually going to cover something, a new sort of approach we're taking to sort of further the studies on diet restriction. And it stems from this idea that despite us knowing about diet restriction for so long and so much research on it, why doesn't everybody do it? And the answer is simple. It's hard. We're not hardwired to do this. And we know this is true because if you look, like I was mentioning, if you look at despite all the increase in our understanding, the rates of obesity are going up. So it's a constant battle with what I think a lot of what I guess sort of the food industry is putting out, such that the food is becoming more and more addictive around us. And this is leading to this increased rates of obesity, and clinical obesity defined by a BMI of 30 is roughly around 40% now, and is expected to reach around 50% by 2030. And almost about 2/3 of the people are considered overweight in America. So, and this can largely be ascribed, I would say, to the amount of sugar consumption, carbohydrate consumption that is taking place. So if you see how much we were eating in the 1700s and the 1800s, I mean, we're almost eating, you know, 10 to 50 times more sugar per capita now than we were eating then. And this sort of mirrors the sort of increase in obesity that we are seeing. I mean, it's sort of also interesting, you know, maybe we can tolerate this much amount of sugar, but after that, the risk of obesity just sort of skyrockets. And why is this so? We think this may in part be due to what we call the thrifty gene hypothesis. And the idea is that we evolved during periods of food scarcity. Now, under these conditions, our system is adapted so that we would assimilate nutrients. So if let's say you encountered a fruit tree, you don't want to just stop at one fruit, for example. You would eat more because you want to ensure that you don't know when your next meal is, whether it's secure, right? So I think food addiction perhaps had a survival value because people who would take more are more likely to survive. And similarly, people who would take in more and store more would be more likely to survive. But it becomes a problem under the current situation where we all have created a secure pipeline for sort of presence of food, and now having these sort of these tendencies to overeat aren't beneficial anymore. So further on this, we think it's not sugar that's really driving this phenotype. Our hypothesis is that what sugar is not as toxic, as reactive per se. So we think it's downstream products of sugar, especially methyl glyoxal, which I'll show in a minute where it comes from, which is a much more reactive molecule, and that forms advanced glycation end products. This, we think, is what's driving aging and a number of diabetic complications, and also doubles the risk of dementia, increases the risk of stroke, cardiovascular disease, and also a lot of evidence argues the role of ages in diabetic nephropathy, retinopathy, and neuropathy. So our working hypothesis is that glucose, when metabolized through methyl glyoxal, kind of is driving aging. And I want to just remind people what this is. For that, you may have heard of methyl glyoxal, maybe the first time, but you're all aware of advanced glycation end products because it's the most commonly practiced chemical reaction by humans. Every time you're cooking, those things that are driving flavor in the food. For example, if you made a toast this morning, just the act of making a toast creates 50 new flavors in bread. So just, sugars and amino acids are reacting in ways, in the presence of heat, dry heat, you get this sort of browning reaction, which makes the food tasty. So this was discovered by Maillard, and it's also known as a Maillard reaction. And initially, it was used to sort of create flavors of meat. So you don't need to eat meat to actually have meat flavor. You could just generate it from specific proteins and amino acids. And you can generate similar experiments are done on us every day. So if you're eating cereal, for example, that is toasted perfectly, and millions of dollars are spent to get our cereal just right using the same chemistry of Maillard reactions. So unfortunately, with age, we're slowly becoming toast as well. And what's happening is that every cell that uses glucose, it divides it into, it breaks it down into these 2-3 carbon atoms, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This is converted by an enzyme, triose phosphate isomerase, and a knockout of this enzyme actually leads to neurodegeneration. So when there's more dihydroxyacetone phosphate, DHAP, a small part leaks as methyl glyoxal. We don't quite understand the factors that drive this leakage right now. So, but I think we think it is important because every cell, from bacteria to us, has created a number of detoxification enzymes, more than what's shown here. Glyoxylases, for example, what they're called, whose job is to basically turn this methyl glyoxal into D-lactate or some other form that is not as toxic, essentially. And what happens, unfortunately, is with age and with diabetes and some people who are more susceptible to this may not have an optimal glyoxylase system. Their returns, it attacks proteins, especially lysines and arginines, and gets cross-linked. And what you're seeing is some of the cross-links formed due to methyl glyoxal. And these are found in our serum, in our cerebrospinal fluid. And normally the kidney clears it. But if all this, if the balance shifts towards more glycation, then the idea is that this drives phenotypes like nephropathy, neuropathy, and even in patients with Alzheimer's and Parkinson's, they've shown that the levels of glycated and advanced glycation end products is almost 2- to 4-fold higher than the normal people. So we are focusing on only one type of advanced glycation end products because we think this is an important one, especially because of our interest in glycolysis and how this is sort of a key sort of and some of the ages that are made are some of the most prevalent ages in our serum. So I'm not going to get into this paper that we previously published in detail, but we identified that methyl glyoxal is sensed by TRPA1 channel. This was all done in C. elegans and some mammalian data is there. But essentially this TRPA channel acts as a sensor and activates SKIN1, or also called NERV2 in mammals, sort of to detoxify methyl glyoxal, essentially. So we think this is part of a feedback reaction where MGO activates TRPA1 channel to increase the levels of these glyoxylases to detoxify them. And these glyoxylases are important because polymorphisms in these glyoxylases have already been implicated in Parkinson's disease and diabetic complications. So for example, DJ1 PARK7 here, a well-known gene for early onset disease, early on Parkinson's disease, is a glyoxylase as well, amongst other functions. So we had, I should say, we identified a couple of drugs, podocarpic acid and lipoic acid, to activate this TRPA1 channel. And this worked really well because we could show that preconditioning a cell with these drugs actually activates the system and protects the animal from methyl glyoxal toxicity. But so this was done using an assay where we measured the neurite length of the neurons and at different doses. And what we did then was try to see if you could add other compounds to further improve this. So from our screen in C. elegans, we had identified about 15 other compounds from a screen and about 800 compounds to improve function. So to reduce methyl glyoxal induced toxicity. So we combined them, and what Jessica Ramirez, a master student, spent 2 years doing this. She tried hundreds of combinations basically to test which ones would work. And what she found was that if you use these 5 compounds together, as you can see here, now these animals are super protected against methyl glyoxal. So you can see here, just when even at the lowest doses, it's protected. And I want to say one important thing about this experiment. In this pink, what you're seeing, so in red is lipoic acid at 1x dose. In pink, what you're seeing is each of the 5 compounds at 1/5 of the dose. Now, even when we're using less, what we're finding is that it's much more protected. So this encouraged us. So this is all in cell culture. So we said, okay, let's see what happens in mice. So we showed that if you give this to mice, you can reduce methyl glyoxal, as shown here. And you can also reduce the downstream advanced glycation end product, MGH1, which is shown here. And as we gave this to the animals, we found, interestingly, that it reduces their body weight, and they're actually eating less food, slightly less food. I mean, about a reduction about, I think, 15 to 20% in their calories. But it has a dramatic effect on their food, on their body weight. So with age, they're not quite gaining the body weight like a normal mouse. And then we asked if this improves their glucose tolerance, and yes, it does. So this is not a diabetic mouse, a normal mouse. And you can see even a normal mouse, when it takes Gly-Low, which stands for GLYcation Lowering, the combination of the 5 compounds, they have improved glucose tolerance. And we could show that their insulin sensitivity is improved. We give a bolus of insulin, and you can see it can bring down glucose even further in these animals. Next, because we were interested in what's happening in the brain, in the hypothalamus, we looked at gene RNA-seq analysis of the hypothalamus, showed that actually very interestingly, a number of genes in the glycolysis pathway are down, except 2, which actually would favor a reduced production of methyl glyoxal, TPI and PGK1. So you want to have these more, because that would favor reduced DHAP and less leakage, we think. So the rest of the enzymes are downregulated, which is very interesting because you want to reduce your glycolysis. And then we found another interesting thing, that not only does it reduce glycolysis, it actually increases more flux through the pentose phosphate pathway. And this is important because this is important for the production of glutathione and also some nucleotides. So now the cell, at least in the hypothalamus, is not undertaking too much glycolysis, but instead is making more glutathione and reducing the usage of glucose. And we found that not only that, but also a number of glyoxylases, which are shown here, are actually upregulated when treated with these compounds. And I'm not going to get into data, but we've shown that this is potentially through the action of this transcription factor NRF2. We also gave this combination supplement then to leptin receptor deficient mice. And you can see we did this at 3 different doses, but you can see a reduction in body weight at the highest dose, quite significant. And what was remarkable was this lifespan improvement in sort of lifespan. So half the animals died in about 6 months, but not a single animal died when kept on the supplement. Next, we asked, what's happening to the gene expression in the hypothalamus? This is not like a normal calorically restricted animal. And we found that a lot of genes actually in the brain are going in the opposite direction. So if you look around this axis, so this is changes in gene expression of caloric restriction on the y-axis, and on the x-axis changes in Gly-Low. So if caloric restriction was the same as Gly-Low, you would see everything across these 2 quadrants. But instead, we see some that are going in the opposite direction. So got interested in what this is. and we found that they're actually surprisingly a lot of genes important for ribosomal translation. We try to understand what's going on, and I'm not going to go through all the experiments, but basically we showed that these normally on caloric restriction, AMPK levels go up in the hypothalamus, and this reduces the TOR activation and S6 phosphorylation and reduces ribosomal translation. We found that instead on Gly-Low, actually, the hypothalamus becomes unresponsive to ghrelin. So we gave ghrelin and the animals don't overeat. We found that AMPK levels are actually decreased and TOR is surprisingly upregulated in the hypothalamus. But that's very interesting because just driving TOR, if you reduce TOR activity in the hypothalamus, it makes the animal overeat. So this is kind of beneficial to the animal to have increased TOR, because as far as signaling goes, it will drive a response so that animals are eating less. But also I want to say the hypothalamus, if you look at the overall gene expression, actually looks younger. So we have a paradox that despite TOR being sort of upregulated in this Gly-Low condition, they feel satiated, but their hypothalamus is actually younger. So then we measure lifespan, given all this sort of data. We said, and we took animals much later in age, and actually we're doing experiments to see if you get larger lifespan extensions than done earlier. So we started at 24 months of age, and you can see, this is about 40 animals each, that mice on Gly-Low condition actually live longer.
Dr. Kapahi, sorry to interrupt. I'm just watching the time. Do you want to wrap up so we have time for questions?
Yeah, I'm finished. Yeah, this is the last slide. And so we show that Gly-Low works in a leptin-independent manner to reduce mTOR food intake and improves insulin sensitivity and extends lifespan. I'm not going to go into this data, but we have some really exciting data, and I'd love to collaborate with anybody who's interested in this diet. We can provide the diet for people who want to do these experiments. But we're seeing improved eye function, muscle function, and really striking reversal of menopause conditions and reduction of FSH, and sort of reduction in sort of ovarian aging. And recent experiments are showing that we can reverse the memory loss that is seen in models of Alzheimer's disease. So with that, I'll just end. And I say, in addition to sort of doing more mouse experiments, we're also interested in sort of clinical trials. And if again, any clinicians out there today, we would love to, I'd love to chat with you to see the possibility of taking this in humans. And I'd like to thank my team and the NIH and AFAR for funding. Thank you very much.
Thank you. It was an excellent presentation. Very interesting talk. Let's answer some questions. A reminder to attendees, you can enter your questions in the Q&A box. Our first question from Larissa Constantou: Could ketogenic diet lessen glycation reactions in the body?
Absolutely. I think that's one of the things people don't talk about ketogenic diet is ketogenic diet doesn't work without reducing carbs. And when you're reducing carbs, I think it's glycation that's affected. And also we're working on another idea, which is out there in the literature, that actually methyl glyoxal directly binds the ketones and actually buffers it. So we think AGEs and ketones definitely have an important role to play and work together. And some data I'm not going to show today, but I do want to say, I'm not sure being on a ketogenic diet is the right way to go, because you also end up reducing glycogen. But definitely low carbs diet is an important thing to have, but I would not suggest people to go on the more Atkins-like diet, which is high protein and low sugar, which tends to put a lot of stress on the kidney and the ketones. So we think we want to reduce the toxic glycation products to be reduce them at the same time, sort of reducing glycolysis. That would also be beneficial. And Chris Wiley has a question, and the question is, it's great to see how these stories are forming a compelling narrative. Thanks. Yeah. Then the question is, has anybody done a comparative lifespan study on blind species, cave dwellers versus closely related sighted species? And also with regard to glycation data, do you see any protection of genomic data? So to your first question, I think that's an excellent question. very interested in this. We don't know. No, those experiments have not been done. But I actually the closest that comes that happens is people use these fish, which especially in like they're in this. There are these fish in Mexico. I'm not blanking on the name, but the cave fish are blind, and the ones that are in the river are surface fish are actually not blind. And what's interesting is the cave fish have been used in zoos and have really long lifespans, and they are actually blind. So I think that's the closest I can think. Chris has done an experiment, but not a proper experiment, but it's a sort of data people have gotten from zoos. And secondly, yes, glycation in MGO does cause DNA damage, and we think the glycation lowering protects against genomic DNA. Another question is by Ryan.
Yeah.
Yeah. What makes?
I can read it. Oh, go ahead.
Yeah. What makes Gly-Low combination unique? Aren't most of the compounds ubiquitous in higher level organisms? Actually, that's a very good question. Yeah, maybe it didn't come across in the talk. We are not using these compounds at what would be sort of in a what you would call a multivitamin tablet, right? These compounds are being used at much higher doses. So this is, I think the idea is that these same vitamins at higher doses, like lipoic acid is given for diabetic complications, already approved in many countries. They have then protective effects to sort of improve homeostasis. And we think the combination at these higher doses, now you don't need as high doses with this combination, but I think using them at a higher dose can be protective. And this, I think, is better than having nicotinamide alone, we think, or lipoic acid alone. You know, thiamine is neuroprotective in many Alzheimer's studies and in clinical trials. So we think the combination could be better, could be much better than the single compound, or which is, there are almost hundreds of clinical trials on these compounds alone. But we think the combination is could provide unique sort of benefits.
Great, thank you. I have one more question. Can you say more about the risk of multimorbidity, how it relates to dietary restriction? Have you looked at correlation of genes other than cognition-related diseases with aging?
Yeah, multimorbidity. Yeah. So in our, you mean in the context of glycation or dietary restriction or what? So
dietary restriction, I think.
Yeah. So dietary restriction, like, is very well covered in our review. Like I said, showed that slide. You know, you see improvement in brain function, gut health, liver function, but I think some things you compromise, like I said, wound healing, bone health in some cases. With regards to Gly-Low, what we are finding also is there's improvement in multiple morbidities like eye health, cardiovascular function, arterial stiffness, dementia. But, you know, we haven't done wound healing. We're working with a collaborator to see how that would work. So we're looking at different morbidity, but all of them don't show equally, you know, equal protection. So we're going through them one by one to see what's happening. Yeah. But that's an important question. But overall, I would say there's no reason to assume everything is improved. People really have to.
I think the end of your answer just got cut off a little bit. We're having some trouble with the audio connection. But I think you were almost done with the question. So our speaker might come back, but since that was the last question, that actually concludes our seminar and we're up on time. So thank you. Hopefully you can hear me. Thank you to the speaker for our talk today, and thank you for everyone attending. We'll see you next time. Bye.